Hello everyone, welcome back. This is our final mini lecture for chapter 3. This is 3.4 and 3.5, bacterial internal structure and the archaea. There are 5 learning outcomes or objectives that you need to master by the end of this mini lecture. First, name at least 4 structures that may be contained in bacterial cytoplasm. Describe the sporulation cycle, including spore formation and germination. Name 2 characteristics of archaea. Identify the structure that is more similar to eukaryotes. Name 3 distinct characteristics of archaea. So this mini lecture, we're going to focus on the internal structures in bacteria, and there aren't many because they don't have membrane-bound organelles. So first is the cytoplasm, and the cytoplasm is about 70 to 80% water, and it's also includes a mixture of a number of sugars, amino acids, salts, and other nutrients that the cell needs. It also serves as a pool for building blocks for cell synthesis, sources of energy, all of the molecules and materials that the cell needs to repair itself or create new organelles, create proteins, create complex sugars are all found within that cytoplasm. The cytoskeleton is also an important component of the internal cell structure, and it helps to create and maintain the cell's shape. Some bacteria produce very long polymers of protein that are similar to eukaryotic actin filaments that compose their cytoskeleton. As I said before, it helps to arrange shape. They are in sort of this helix shape. They have also been identified in archaea, and they can be a target for some antibiotics in order to address infections and bacterial disease. Another component of the internal structure is the genetic material in the chromosomes. Remember, there isn't a nucleus in bacterial cells, so instead we have this general space or area within the cell called the nucleoid, and this is where we find the bacterial chromosome. It is circular instead of linear, and it is composed of DNA. Some bacteria also have small circular pieces of DNA that are found within the cytoplasm called plasmids. And plasmids typically do not contain essential genes, but they do frequently contain genes that code for things like drug resistance, toxin production, production of different enzymes, and they tend to increase an organism's virulence or ability to cause disease. Another internal structure that we're going to find in bacteria and archaea are ribosomes. And ribosomes are the site of protein synthesis, and they are composed of ribosomal RNA and proteins. And there are 2 parts of the ribosome. There is a small subunit and a large subunit. And when we put them together, that is this total S or measurement of size. So bacterial ribosomes are 70S. They are composed of a 50 subunit and a 30 subunit, which ends up totaling 70S. I know 50 and 30 is 80, but somehow when you combine them, it ends up with this total measured unit of 70S, whereas eukaryotic ribosomes are 80S, and it's a 50 unit and a 40 unit, if I recall correctly. Archaeal ribosomes are also 70S. However, their ribosomal RNA and their subunit components are more similar to eukaryotic ribosomes than bacterial ribosomes. Some other structures that we might find inside bacterial cells are inclusion bodies, and these structures can be used for a number of things. Food storage, they can also be used to contain gases to increase an organism's buoyancy. That's particularly useful if the organism tends to live in an aquatic environment. They can also store crystals of iron oxide that provide the organism with some magnetic properties. There are also things like microcompartments that contain enzymes that are designed to work in some of the biochemical pathways within the cytoplasm of the bacteria. Finally, we also have things called endospores. And endospores are not produced by all bacteria, but they are produced by some. And they are dormant bodies that provide or increase, I should say, the survivability of a particular organism if conditions within the environment become too drastic for the organism to survive. A number of organisms produce these, including Clostridia, Bacillus, and Spirocina. The vegetative cell is the living, metabolically active cell, and then we have sporulation, and this occurs when conditions in the environment change so that the organism can then survive. And we're going to talk about this whole process. So endospores allow an organism to resist extremes like heat, drying, freezing, radiation, and other harmful chemicals that would kill a cell, acid, that kind of thing. This is the germination process, how the spore is formed, and then how sporulation occurs. So we begin with the vegetative cell. This is the living or the metabolically active cell. And once the cell detects that the environment is changing and not conducive to its survival, it first duplicates the chromosome and separates that chromosome into two parts of the cell. So we have this forespore and then the sporangium, and each one contains an exact copy of the DNA. The sporangium is going to engulf or swallow the forespore, and then the sporangium begins to produce and create the layers that are going to form the spore. So we have the spore developing here within the sporangium. These layers continue to build until we have this outer cortex that is developed, and we finally get this mature spore. And the DNA within the sporangium then is going to begin to break down, so that what we have left is the spore, contained in several different layers that are going to protect the spore from any changes in the external environment. Once the spore is in an environment where the cell can survive, so the heat has been reduced or water has become present or the pH has changed, then the spore germinates and it swells. These outer layers rupture and the vegetative cell is released. So this is the process of germination and sporulation. Okay. There are a number of medically significant bacteria that produce spores. Bacillus anthracis is the agent of anthrax. It is spore forming. all of the Clostridium organisms, tetani, perfringens, botulinum, and difficile produce spores, and they cause some pretty nasty diseases. So let's talk about archaea really quickly. Archaea are the other, I guess, prokaryotes. They are considered a third cell type, and they are all included in a separate superkingdom. Scientists have determined that they are more closely related to eukarya than bacteria, as we saw in chapter 1 with the phylogenetic tree. And there are some key structures that show this similarity, and the first is in ribosomal RNA. Ribosomal RNA in archaea is much more similar to eukaryotes than bacteria. There are RNA sequences found in archaea that are not found in bacteria. Also, the process of protein synthesis, or building and forming proteins within archaea, follows much more similarly to the pathway that eukaryotes follow in order to form proteins than how bacteria produce proteins. And the ribosomal subunit structures are more similar as well. Archaea, we talked about, they have unique RNA sequences. They also exhibit a unique method of DNA compaction. And they contain unique membrane lipids, cell wall components, and pilin proteins. Archaea are also considered extremophiles. A large number of them are found in some of the most extreme environments on Earth. They can be found in extremely high or low temperatures, extremely high salt or acid concentrations, and some live on sulfur or methane and actually use sulfur or methane as their means to produce energy. Some of them do live on and in the human body and can be capable of causing human disease. So this table is actually from your text, and it compares the three cell types, bacteria, archaea, and eukarya. You can see that both bacteria and archaea have single chromosomes that are circular. We have 70S ribosomal structure, although the structure of archaea ribosomes is much more similar to eukarya. All three cell types contain unique ribosomal signature sequences. Protein synthesis is similar to eukarya in the archaea. Bacteria produce proteins in a unique way to eukarya and archaea. Peptidoglycan can be found in the cell wall of bacteria, but not archaea or eukarya. Fatty acids are found within the cell membrane of bacteria as well as eukarya. However, archaea have long chain branched hydrocarbons in their cell membrane, not fatty acids. Sterols are found in the membrane, only in eukarya. Nucleus and membrane-bound organelles, also only in eukarya. And then there are flagellum found in all three cell types. However, the flagella in eukarya and the flagella in archaea are unique to the flagella in bacteria. And that is the end of 3.4 and 3.5. Check that you can answer these questions and that you understand the concepts from chapter 3. Remember, you can go back and review these videos, and hopefully it was helpful.